This method can help answer key questions in the chronic lymphocytic leukemia field, such as the accurate prognosis of patients. The main advantage of this technique is that it is simple, fast, and highly accurate.
The implications of this technique extend towards robust prognostication, most particularly risk stratifications of patients with chronic lymphocytic leukemia, because the accurate prognostication may lead a refined therapeutic management of the disease. Visual demonstration of this method is critical, such as the PCR product gel excision and IG sequence analysis steps are difficult to learn because they require both precision and experience.
To begin this procedure, add 200 microliters of EDTA to a 50 milliliter sterile collection tube. Add 20 milliliters of peripheral blood and 15 milliliters of RPMI medium to the tube, and pipette up and down 2 to 3 times to mix. Next, add 15 milliliters of density gradient medium to a new 50 milliliter collection tube. Slowly layer the peripheral blood solution onto the density gradient medium, making sure that it does not disrupt the gradient.
Centrifuge at 800 times G for 20 minutes with the centrifuge brake off. Then use a sterile Pasteur pipette to collect the mononuclear cell ring that has formed between the upper plasma platelet layer and the density gradient medium layer, and transfer it to a new sterile 15 milliliter collection tube.